Good day everyone, welcome to the course on biological data analysis and visualization with R. So, in the last few classes we have looked into some of the visualization packages like ggplot2, ggscatter, etc. And then we also talked about the data organization packages, data transformation packages including tidr and dplyr. In this class and the subsequent class, we will be talking about bioconductor packages. So, what I will be doing is first I will give you an overview of the bioconductor packages, what are the packages that are there which could be useful. There are a large number of packages, and what we will do is we will take some of them in this class and the next class, right. We will discuss two packages, and with hands on we will see how these packages could be useful. So, that is the agenda for this class. So, first I will show you the list of bioconductor packages and we will get some overview of these packages that could be useful for us, and then one package that we will be talking about which actually helps us connect to biological databases, very important databases in R, right. When we are doing this programming, data analysis, statistical analysis, we want to connect to these biological databases, right. For example, we need some information including, for example, gene ontology, gene function, etc. So, those informations we can get so from R itself, bioconductor package itself. Okay. So, we will talk about one such package in this class, and then in the subsequent class we will talk about another such package, and we will apply again to a data set, and with the help of hands on we will kind of illustrate how these packages could be useful. Of course, as you realize, there are a huge number of packages, so it is not possible to discuss all of them and kind of give examples for all of them, but what we will do is we will try to cover at least some of these in these two classes. And then followed by that, when you actually go into later classes, we will extensively utilize a lot of these R and bioconductor packages for statistical analysis. For example, we will be talking about WGCNA analysis or ChIP-seq data analysis. In those cases, we will use many of these bioconductor packages. Okay, so let us begin first with the bioconductor packages. So, these are the set of packages that are specifically built for bioinformatic analysis, and these are here, right. And we can classify some of these packages according to their kind of applications. This is of course a broad classification, and there is no formal classification, but if you go through the packages, you will see, for example, some of these packages can be applied in specific biological experiments, right. So, you can see, right, these experiments, for example, you have done some experiments and you can apply these packages. You also have packages that actually have statistical methods, specific statistical methods that are that can be applied with these packages, and there are some packages that actually summarize workflows, right. So, how do you actually analyze specific type of data for through different packages and so on. Okay, so, for example, we have some of these packages in bioconductor called dsec2 or AGR for differential gene expression analysis. Of course, I will not be discussing that. This I have discussed in an earlier course. And there are other packages like LIMA-FE. These are also used for microarray experiments. Okay. So, if you are doing microarray experiments, these packages could be interesting for you. You also have specific packages that are actually data formats or containers, right. So, in addition to the data formats that we talked about, data structure that we talked about, many of these packages they require or define their own data format. Okay. And these are the common data formats that are often defined. For example, if you are working with single cell data, single cell gene expression data, for single cell experiment could be useful, or you can have spatial experiment, single cell allele experiment, etc. So, we will explore this a bit once we go into the bioconductor web page. We will see the list of packages there. We can also have databases or database connectors, right, that could be useful. For example, we have Genome InfoDB, we have Biomark, and then you have these packages organism hs.eg.db. Again, this is organism specific databases. So, HS stands for Homo sapiens. So, this is a human database, right, data for human data. Then you have MM, right, which is for mouse, etc. You can find this across many organisms. Okay, these are built-in packages in R. So again, you can load these packages in R and you can connect to data and you can use those data for your analysis. Similarly, you have like you can have RTrack layer or cake graph. These are the packages that actually help in connecting again to different data, different types of data, right, and this could be directly used in our data analysis. So we'll see in a moment, and I'll also show you, right, how we can explore and what are the functions of these packages, what are the commands that that you have in each of these packages. This can also be looked at. Okay, so for the hands-on, we'll take this Biomart package, right, and very briefly I'll describe what this Biomart package is, and we'll also see what Biomart contains. And with an hands-on data set, we'll also build on this Biomart data. There is another database we'll also see again very briefly is called geo.db, right. So again, gene ontology. So some of you might be familiar with gene ontology, right, what it contains, right. So it contains biological process or molecular function associated with genes, right. So for each gene, so each gene performs a function in a cell, and you have this information or association stored in there in gene ontology, and this can also be used here from this geo.db package. Okay, so with that very brief introduction, let's now dive into the Bioconductor web page first, and then we'll look into the hands-on. Okay, so I'm first going to the Bioconductor. Right, here is the Bioconductor page, right. So Bioconductor home, as you can see, right, this is where we have all the tools. So here, if you just click on this, here you will have the list of packages, and you can see a lot of this. Okay, so for example, you can see some of these kind of mentioned here, different types of packages, right. For example, you can say statistical method, right, or you can say workflow step packages, right. So under this you will have some certain packages classified, etc. Okay, so let's look into all packages, right. Kind of there are about 2000 packages. So I'll just very briefly scroll you through this, and again here is kind of like the rank, right, how heavily used these packages are usually, right. So you can see many of these packages here, and what you have is the package name, right, and the maintainer and the title, and then the rank. So this title gives you a brief idea, gives us a brief idea about what this package does. Okay, and this is something that could maybe perhaps when we are looking for certain applications or certain analysis process, we might be able to search for these applications here, and then we can choose the appropriate package from this title, right. So this is something this is possible, right. So as I said, right, you have, for example, single cell experiment package that I mentioned. So this is for single cell data, right. So this is S4 classes for single cell data, and then you have for example HDF5 array, right. This is mostly a hierarchical data format, right, and with a specific structure. Then you also have other functions that, for example, we use this gene filter function. Again, in subsequent classes, one of the classes will use this gene filter function for our analysis. There are other packages, again, a lot of them, of course. Some of them could be useful for you. So what I'll do is maybe I'll just first let's see if we can find the Biomart package, right. Okay. So here are some packages you can probably see. The AJAR is there, DSEC2 is there, right. These are the packages used for differential expression analysis. Then you have the Biomart package. I'll open it, and we also have, for example, the RTrack layer. So as you can see, Biomart and RTrack layer, what is there? They are actually interface to some databases, right. So Biomart is an interface to Biomart database of Ensemble, right. So you can have a lot of information, like for example genome information from Ensemble, for example, you can get through this Biomart. Okay. Similarly, you have RTrack layer. This is, you can get genome annotation files from the UCSC genome browser, right. So through R, right, you can actually access these databases. Okay. Beyond this, there are a lot of other packages. Again, of course, we cannot discuss all of them. All right, so let's see the Biomart now, right? So here is a collection of databases, right? So for example, Ensemble is one such example. So you can actually load data using this package from these databases, and the one that we will use is for Ensemble. Okay, so Ensemble information, right? Ensemble genome browser, we can actually access this, right? So for installation, this is how it works, right? We have discussed this before, how you install a Bioconductor package, and here you have, for example, the reference manual, right? Accessing Ensemble annotation with Biomart, right? And also you have this using Biomart other than Ensemble. Okay, so here are the examples in much more detail how you should use, right? Here you can see library Biomart, Ensemble, which Biomart you want to use, and then data set that you want to access, etc. Okay, so you can also say list Ensemble, right? And it will give you what are the you have, what are the databases are there, and then how do you choose a data set? You can say list data sets, etc. So all these things are written very nicely here with all the details and etc. with detailed description of all this. Then you also have this list of archives, right? So this is something that you see sometimes we need to, we can, when you want to access this database, we have to say either http www.ensemble.org, or you can directly refer to these archive versions. Okay, so there are different archive versions here. You can see as early as like 2009, etc. So sometimes these archive versions could be useful because some of the data sets that we had, or sometimes we look at some data that actually use old Ensemble annotations. Okay, so that's why we may have to use this archive, right? So anyway, take a look, right, how you actually can use this. So, and this is kind of very clearly described here, and how to build this query, etc. all these things. Okay, so we'll see some of this in a moment, right, when you go into the hands-on part. Okay, so here if you want, we also have how do you actually access other databases, like OneBase for example, using Biomart. So this description is there. Okay, so for our application, for our purpose, this is enough because we will mostly use for Ensemble, and this is what we are going to use. Okay, so I just wanted to also show you this Ensemble gene browser. Okay, so here we have this information and annotation, right, which we can use for our purpose, right. So we have this annotation. Okay, so let's now go into the hands-on part, right. Let's see, and as we go into hands-on, we again have an R code. So the first thing we'll do is we'll first load the Biomart. Again, if it is installed, that's good. If not, you can, yeah. So I think this is the so you have to look into the spelling carefully, right, whether it's capital or small. Okay, so this is fine, and I'll also load geo.db. Okay, so it's saying it's masked some of the functions from dplyr, etc. It's okay. So we have loaded these libraries. Now we need a data set. Of course, this data set is here already, right. Let's see where we are. Okay, so we are in the right place. So the data set is called process GSM dropsec data, right. So this is a single cell RNA sequencing data, publicly available, that we are going to use for this illustration. Okay, so I just load this data. I just load this data here, and it's loading. It will take a bit of time. It's a big data set. So what I want to show you, might ask, like, I would make this data available, but you might ask, like, how did I get this data? Okay, where did I get this data? So this number, as you can see, is actually gives us an ID, and this ID comes from NCBI Geo. Okay, so very briefly, here is the NCBI Geo. We can click on this. Let's see, and here you can simply search this ID, and it will give us the data set. Okay, so here is the full data set that we are going to use, and we have downloaded this data. Okay, so and it's a 1.9 MB. It also describes the data, right, what kind of experiment has been done and how the data was processed, etc. All these things are in here. Okay. So, this is a Drosophila data set. Here it mentions this Drosophila, right. So, Drosophila melanogaster. This information is important because we have to use the appropriate data set, appropriate genes, right, gene symbols, right, from Drosophila and from ensemble. Okay, so we are also loading this Go map, right. This is the gene ontology map. So it is giving me some warning, so that's okay. There is no error, and because we'll use this Go map for actually generating the Go term list, okay, map list. Okay, so how do you generate this Go map? There is a function. Again, this function is written later. I'll show you and how we actually create this from this GoDB. Right, so go.db is there, right, this library. We can build this Go map or go mapping.txt. This is the file already present, but we can build this from GoDB file. I will show you in a moment. So what we are doing is we are taking this data and we are taking this count data and storing it as a matrix. Okay, why? Because this is something that will be useful for Biomart, right? So it will take data in matrix format. Okay, so with that we have loaded the data. So the next step is to actually now use the Biomart and use the ensemble. Okay, so as we seen, right, these are the commands. So we have so these are the commands, right. We have seen, right, how do you access ensemble, right. This kind of tells you how do you actually use ensemble, right. So this is the command we have to do, right. We have to use and we have to execute here. Okay, so ensemble use mart. Okay, ensemble mart ensemble data set is D melanogaster. So as I said, this is Drosophila data set. So we have to use Drosophila gene information from ensemble, and host is, as I showed you some of these archives. So I am taking one of the recent archives here to store to load this data. Okay, so we connect to the so this Biomart, right, it will connect to that database, to this website, and it will retrieve this data, right, Drosophila melanogaster gene ensemble. Okay, so this is now loaded. So what we can do is we can look into this, what kind of information we have here. Okay, list attributes. So this is the command to see what kind of information is there. So first 10 rows. So here are the names of these features or the attributes that are loaded now. So first is name. We have ensemble gene ID, right. For every gene we have a specific gene ID. You have transcript ID, etc. You have chromosome name, start position, end position, strand, etc. So all these things are there. Similarly for description, right. So this actually description means it is describing what this name is storing, right. It's showing gene stable ID, transcript ID, etc. Gene start, right, base pair, gene end, etc. Strand means positive strand, negative strand, etc. and karyotype band. Okay, so this kind of information is there. Okay, we can see what we also have transcription start site, etc. Okay, so we have seen these attributes. Now the next command is, what we'll do is we'll get this, right. What we'll do is we'll get this ensemble gene ID and go ID, right, from these attributes we want from there, right, from Biomart, and the filters is on ensemble gene ID. Okay, and values would be row names equal to CD and mart equals to ensemble. So ensemble database we have loaded already. So this is the database we are going to use. Values is row name CD, means we are going to input this information or get this information from ensemble only for genes in this database. Okay, so maybe I have not showed you what this gene looks like or how the CD, the count data looks like, right. Okay, so this is how it looks like. So here the rows are the gene names, fly based gene names, and the columns are the samples. Okay, so we have this gene versus sample, and then what we have are the counts data, right, gene expression count data for every gene here, right. So this is the data, and this command is saying, okay, take only those genes which are present in this list in this data set that we had loaded, and filter by ensemble gene ID, and attributes that we want to get is ensemble gene ID and the geo ID or gene ontology ID. Okay, so this is the geo we have built, and then what we want to get is geo term. Okay, so geo term is term go ID, right. So these are the geo terms that are associated with these genes. Okay. Okay. For example, the ATP binding, etc. Right. So head geo, right. So here for head geo, this is the data that we got from Biomart. We have ensemble gene ID, right. This is the gene ID. Then we have the gene ontology ID. Then you have the term, right, some function, ATP binding, cell development, cellular component organization, etc. Okay. And then we have this, these are the terms, right. So we have this gene to function association. Okay. What kind of function they are related to. Okay. So that term we have also got added here, right. That is given, that is stored as geo term, right. So this term is there. Okay. So now what we do next is we create this co-environment, right. And what we are doing is we are splitting, right. So we have this, we put this together, right, and then we split. Okay. So what we'll do again, we'll look into co dot environment. Okay. So what we have is for some genes, right, we have for some terms, right. So this is NA term, right. For some terms, right, here is the mitochondrial inheritance. This is the term, these are the genes that are under this term. Okay. So they are all associated with this term. So we are kind of reorganizing the whole thing. Okay. And finally, we can create this map list. So when you look into gene ontology, you have three classes, right. You have BP, you have MF, you have CC, right. So BP stands for biological process, MF stands for molecular function, and CC stands for cellular location, right. So cellular localization, cellular component, CC. So what we are interested in here is the only that are associated with biological process. So in that case, we're creating a map list. We're saying from go map, go ID, which go map class is equal to biological process. Okay, and we can see this, what are the go class that are in here. Okay, so we have created this map list, and we can also get a summary of this. And here is the length, right. We have about 12,000 go ID, right, and they are all associated with biological process. Okay, so this is just an example how this ensemble, this Biomart, right, how it allows us to connect to, for example, database like Ensemble, and we can actually get this information annotation. We can also this go terms, go IDs, etc. We can associate them from the go DB as well, and this mapping list could be very, very useful for many different applications, right. So for example, we want to look at functional association of genes that we have identified in certain process, etc. Now what I'll do next, as I said, is we can create this in this hands-on, we actually use this go mapping function, right, go mapping dot text. Now this go mapping text we can actually create ourselves from this go.tb, right. So that's what we, once we create that, we can actually create these maps and we can actually use them for our purpose. Okay. So how do you do this, right? This is how okay, this is how the whole thing works. So we have the library go.db, right, and what we will do is we'll store this first as go terms. So we'll take all the go terms that are there, right, and store as a list. So it will take a bit of time because there are many such go terms, right. So let's see what we have. Okay. Okay, so maybe we could use head function because it's actually a really big data set, right. So yeah, okay, so it's a really huge data set, right. So it's kind of still kind of writing here. Okay, what I'll do is let's say I'll create so that we can continue on this. Okay. Okay, so we have created go.db, and then this is the first command, right? We are reading the Go terms that are there. Okay, so let's just say head XX. So what you get is all the terms that are stored here, right, in Go term. Okay, so we are getting this as a list, and you have the Go ID, you have the term mitochondrial inheritance, ontology is the biological process. Okay, so that's the term we have loaded. Now what we want to do is, okay, so we calculate this. This is the length of this. So we have 42,000 elements in the list. Okay, and what we'll get is, now we are creating now this output. Okay, as a matrix we should have 3 columns and number of rows would be this 44,000. Okay, and they should be NA values, right. So this command actually initiates, right, this matrix, right. This is initialization of the matrix. Now for this, now how do you fill up this matrix is using this command right here. So for I in I 1 colon nn, so I runs from 1 to nn, right, and this is how we run for loop, right. This is something we have seen, right, when we in the introduction. A would be first the Go ID, B would be the term, Go term, and C would be the ontology, and for each I, right, so this is for each row, store these, right, as A, B, C. Okay, these are this is the information that will store here. Okay. Okay, so this information is now stored. What we can do is we can check this output. This is how it looks like, right. So this is the geo term. This is, then we have the geo function, right, the geo term actual function that's describing here, and then you have the biological process or molecular function, right. As we described, there could be 3 types. Now once this is done, this is actually very simple. You can put this together, right, as a data frame. Okay, this double quotes are gone, and we can also have these column names now, right. We can, column names are this Go ID description class, and then we have seen the output. We can simply now write this, okay, into the Go mapping file. Now this Go mapping file we can use, right, we can use this Go mapping file, right, for we read this here, right, in go.map, and then we use that for Go term associations and creating this gene to Go term association, right. So this is something how we utilize, right, because from Ensemble we got the gene ID and the Go term ID, and then we actually associated each gene to this geo term and the function. Okay, so the goal of this, you can see, right, how these databases could be very useful. For example, we have illustrated Biomart, right, we could connect to Ensemble and load data from there, and also we have seen how GeoDB or gene ontology helps us, right. We can actually again use this library to actually create this gene to function association. Okay, so we have illustrated the use of one package here, Biomart, and also we have seen how GeoDB could be useful. So here are the references. So please feel free to explore these tools that are available with Bioconductor. We'll use some of them in our later classes. So to conclude, so we have had an overview of the Bioconductor packages. So we have talked about some of the packages, right, and different types of packages that are available. Of course, this is a really big number, and it's impossible to discuss all of them here, but we tried to pick one or two packages in this class. We'll pick one more package in the next class, and we'll illustrate its utility again in the next class. We also had some hands-on with a database access package, the Biomart, and we have also seen how we can use the GoDB package. Thank you.